Welcome to our podcast, Oncology Morning Commute, the emerging role for antibody-drug conjugates in GI cancers. Morning Commute is developed in collaboration with At Point of Care and Projects and Knowledge and is part of a continuing medical education series. This independent CME-CE activity is supported by an educational grant from AstraZeneca Pharmaceuticals and Daiichi Sankyo Incorporated. In this episode, Dr. John Marshall and Dr. Howard Hochster discuss GI cancers and antibody-drug conjugates. How are these new therapies changing the landscape of GI care? Information about the faculty and disclosures can be found at morningcommutepodcast.com/solid tumor 6. You can use this link to receive your credit and evaluate this program. The URL can also be accessed in the episode notes. Dr. Marshall is a professor and chief of hematology oncology at Georgetown University in Washington, D.C. Dr. Hochster is a distinguished professor of medicine and associate cancer center director of Rutgers Cancer Center in New Brunswick, New Jersey. I am Candace Hoffman, managing editor of Morning Commute. Dr. Hochster will begin our discussion.
To our listeners, thanks for joining. I'm Howard Hochster from the Rutgers Cancer Institute, and I'm here today with John Marshall, one of my good colleagues in GI cancer and someone I always enjoy speaking with. And we were continuing our podcast on the emerging role of ADCs in GI cancer. Welcome, John.
Howard, thanks very much. And thanks for working together on this. Who knows, pretty soon we'll be podcast famous. You never know.
Could be. But I'll always say I knew you when. So I think we had a little discussion earlier. If you missed our first podcast, we talked about the role for HER2 testing in GI cancers. And it's not as frequent as we see in breast cancer, but we still need to test for these. And we've got some good therapeutics now. So one of the main areas that we are interested in now is the evolving role of antibody-drug conjugates, which will take an antibody targeting HER2 and attach a payload to it, kind of a Trojan horse approach. It's got, as John pointed out earlier, the advantage of turning off the cell growth pathway by the antibody and then delivering some kind of cellular toxin to the cell that has the overexpression. We're going to dive a little bit deeper into the therapeutic options and the data behind the approach to HER2 overexpressing GI cancer.
Yeah, I was thinking, Howard, we can't emphasize enough the importance of testing. That if you don't do this, you'll never know about it. These are active therapeutic options for these patients. There are enough of them out there that you really have to test for every patient. There are blood testing, tissue tests that are available for this. And now you've got not only frontline therapies, but also subsequent line therapies. And this is going to, I think, in the end, translate to maybe one day adjuvant, although for right now, we don't have that indication. But anyway, you've got to go here. I know there's a lot of immune therapy fever. So we've gotten a pretty high awareness of PD-L1 testing, but HER2 is equally important, maybe more important than PD-L1 testing, a more powerful predictor for therapeutic benefit.
Right. So couldn't agree more. We need to test and look for these targets. So I think the first trial that really showed the benefit of HER2-directed therapy was the TOGA trial for gastric and GE junction adenocarcinoma. I think the whole stomach, esophagus, GE junction area, especially with the immunotherapy trials, have gotten kind of all conglomerated. So we have some trials that have squamous adenos, esophageal adeno, esophageal squamous, GE junction, gastric, all in one trial. But I personally still feel the need to separate these to a certain extent. And for HER2, we're really looking at adenos of the stomach and the GE junction, not so much the distal esophagus. But the TOGA trial showed that if we add trastuzumab to chemotherapy, we do improve disease-free survival and overall survival. And that's become a standard first line for HER2 overexpressing gastric cancer. So we test all our new gastric cancers today with immunohistochemical testing, kind of as a reflex testing.
Yeah, I was thinking back to, I had hair when that study was done. And, and, it's really taken a long time for there to be significant progress. And it's not without trying, right? We had studies of combinations, we had adjuvant studies, we had second line studies. So we were continuing to borrow successes from the breast cancer world and try to apply it to this gastroesophageal world. And we really failed to move the bar. And it's always caused me to scratch my head as to why there wasn't the carryover, why we weren't seeing the same kind of benefits in our cancers compared to breast cancer. And it almost made me question the validity of HER2 in the beginning.
Right.
Was the TOGA trial a funky outlier that it really wasn't that good of a target? So I'm reassured by the newer data that's come forward today.
Yeah, the strategies of combining trastuzumab with pertuzumab and with TDM1 or use of TDM1 after just did not work out that well in gastric cancer as breast cancer. And I don't know the answers to that. Maybe it's bypass signaling or collateral pathways, and maybe it's just the patient populations that we're doing these studies on. It's rare enough that most of these studies become worldwide studies, and maybe there's heterogeneity in the patient populations. I don't know.
We have improved testing, that's for sure. Testing techniques and standard of care there has improved. And I like to come back to your opening point about that these are different cancers. Is that, are we lumpers or splitters? It's squames, it's adenos, GE junction, and stomachs. And even stomachs come in two flavors, the sort of diffuse and intestinal kinds. And
yeah.
Then we start looking at Asian populations versus Western populations. We see differences in outcome there. And so, we're making some assumptions how these translate or are the same when in fact they probably are clinically distinct. And because they're not all that common in our, populations, we have to make decisions about, lumping patients into clinical trials. We're seeing it with immune therapy now, where we lump them and then after the fact split them back out to see if there were groups that benefited more than others. And I think that may also have been true with some of the bumps that we've had with HER2 targeting over the last decade or more.
Yes. So, one of the newer drugs, which I'm pretty excited about, is an antibody-drug conjugate that's trastuzumab attached to a topoisomerase 1 inhibitor, deruxtecan. And, that's the drug that was in the Destiny gastric trial. So those drugs do have a certain kind of toxicity we need to look for, which includes corneal ulcerations and interstitial lung disease. But the response rates and the survival benefit was really impressive. It was published in New England Journal, and it showed that in a 2-to-1 randomized study of 125 patients, the hazard ratio for survival was 0.6, with a median of 12.5 versus 8.4 months. So a 50% improvement in survival in the gastric trial with the trastuzumab deruxtecan. So I think that that's really one of the better targeted therapies we've seen around.
Yeah, no question. And if you put that in the fact that this was in third line in the clinical trial, even though there's some wiggle room about lines of therapy with our FDA approval, really impressive response rates, but in the 50% range for the trastuzumab deruxtecan arm, and comparing it to chemotherapy on the other side, which performed its typical, say, 10% response rate. So big deltas in response, as you say, overall survival deltas that were impressive and meaningful to our patients. And very quickly on the backs of relatively small clinical trials. I think this is also a sign of the times, is that you don't need thousands of patients to define benefit when the benefit is obvious, when you almost don't need a biostatistician to show the deltas. And so, smaller, highly enriched studies, 2-to-1 randomization, are enough for us all to say, this is worth it for our patients. So I think the Destiny Gastric 01 study is a landmark study, certainly justifies approval for this patient subpopulation in what is a bad cancer, right? These are patients that often don't make it to second and third line because of the nastiness of the disease. So this is a really good option for these patients.
Yeah, and I mean, that was such a well done study. Of course, it led to the FDA and EMEA approval in Europe, and also into the NCCN guidelines last year. So, trastuzumab deruxtecan is a drug that you should be able to access for previously treated HER2 overexpressing gastric cancer that have had trastuzumab in the past. I mean, it's approved and it's in the guidelines. So, and companies continue to do trials. There's a second line trial, again, looking at use just in second line instead of third line. Then there's a randomized trial against what we often give second line, ramucirumab and paclitaxel. So a lot of interesting data will come out with the use of this antibody-drug conjugate.
And my prediction is that it's going to keep marching forward to first line and maybe even adjuvant studies. Fingers crossed that we get some benefit from those. Now, in terms of testing, I think there is not a requirement to repeat testing for these patients, and it kind of comes back to the mechanism of action. They've begun to look at levels of expression of HER2. Now, to be in this study, your baseline test needed to be HER2 3+ or 2+-ish positive to get in. But because this drug is a hook, right, to bring a payload, to bring a chemo payload, there have been some studies that have looked at lower levels of expression. And while not approved in those lower levels of expression, there are some observations of benefit even when the receptor's a little lower on there. So what do you think about that? Do you think we're going to need to dig deeper on those lower expressors and define those groups to see if there's any benefit worth going after?
Yeah, I think so, John. I mean, we don't know these things. And even in breast cancer, some of the large trials, NSABP, for example, have shown some benefit to trastuzumab-based therapy for people who have low or no expression. So, there may be benefit for lower expressors besides the 2+-ish positive patients. And then, of course, there's a real question how to sequence it in with immunotherapy. I was personally very impressed with the Keynote 811 trial. That was the trial looking at first-line therapy with chemotherapy, mostly Capox with trastuzumab, and then Pembro or placebo. And it was interestingly published in Nature, not even a clinical journal. But the primary endpoint for that was the response rate. So the response rate was basically 75 versus 50%, more or less, for the addition of the pembrolizumab, and the FDA approved it based on that. I'm looking forward to seeing the PFS and survival benefit also from that trial. But with people who have higher CPS scores, 5 and higher, I'm really much more inclined to use anti-PD-1 therapy early in their treatment, and how to sequence that with HER2 is interesting. Also in the 811 trial, it didn't matter what your PD-1 expression was.
Yeah, no, I think that's an incredible observation. We all know that we've been combining PD-1 inhibitors with everything, just in the off chance that it's going to improve outcome. So there's almost no clinical scenario that I can think of where we haven't at least attempted, regardless of biomarker, to throw in a PD-1 drug and see if it moves the bar at all.
Yeah, I know. There's a lot of, let's take PD-1 with another inactive drug and try to make it work in colon cancer or pancreas. But I mean, fortunately in gastric cancer and maybe biliary cancers too, they actually work. So
yeah, I mean, but even then, you get these smaller deltas that probably are meaningful. And over the years of GI cancer, small deltas is what we've gotten used to. So we'll take it. But I was impressed by the Keynote 811 study, and I thought, it suggests there's some sort of potential mechanistic interaction here versus just additive benefit of IO working in this space, HER2 working in this space. So I do think that study further supports the need to have HER2 testing in first-line therapy. Don't just be thinking your CPS score, be thinking both. So you need to send both. Now, they're both doable with immunohistochemistry with relatively quick turnaround. So the key ones you need in this space can be done locally. You don't need to partner with somebody out there in the world to get those results, and you can have them in place for frontline therapy. So I think it's a practice-changing clinical trial right on a dime.
I agree. And, then we shouldn't also forget there's other small molecule inhibitors. There's the studies going on with tucatinib, both in gastric and colorectal cancer, that may have a role as well. So we have a number of options how to sequence drugs with HER2 overexpressing colon cancer and gastroesophageal cancer. So what's your approach now on HER2 in colon cancer? Are you testing everyone?
Yeah, I mean, I think you have to. As you pointed out in our earlier podcast, it is the RAS wild type, BRAF wild type, left-sided patient that is also frequently HER2. And so if you're going to enrich for a better return, doesn't mean don't test everybody with colorectal cancer, but it's that group that needs testing. And I tell you, that's also the patient that you're tempted to give EGFR therapy to, even in front line. And yet we've got evidence to say that frontline or EGFR targeting in HER2 overexpressors is not as effective. Signs of resistance, maybe even inactive in that group. So I think HER2 it has to be in that initial portfolio of testing of MSI, RAS, RAF, and then HER2. There's some other rarer tests, the Intrex and the like, that could wait till later. But I tend to test our colon cancer patients for everything as soon as I can. And what's nice about colon cancer is usually there's no shortage of tissue. There's often liver biopsies or primaries have been resected where tissue is easily obtainable. But then we have to remember to track it. We have to remember to bring it forward in our HPI so that we remember what is out there. So absolutely testing everybody with colon cancer and tracking those where I haven't yet. You?
Yeah. Unfortunately, there's no HER2-directed targeted therapy that's FDA approved for colon yet. We have some single-arm studies like the MyPathway study for trastuzumab and pertuzumab, and therefore it's in the NCCN guidelines. So, for second or later line therapy, you may be able to get those approved. But the key trial now is the SWOG 1613 that Dr. Raghav is sharing. He's from MD Anderson, works with Scott Kopetz. They've done a lot of the work on this area, showing that it's a bad prognostic factor also. But, once we get that study done for second line versus chemo, hopefully that'll lead to an FDA approval. So, if you have these patients and you have the trial open, don't forget about that. And then, of course, the sponsors for the trastuzumab deruxtecan are doing a colon study. Destiny CRC01. That'll hopefully also have some pretty good data coming out that may also lead to supplemental FDA approval. But, it's a battle to get these drugs approved today. So, we need the data.
Yeah, if we think about, I don't know what the actual number of all comers in colorectal, I think of it as more like 2 to 3% at most. But when you start to enrich for this all wild type group, you start to see them. And that's then becomes a patient base where we really need these therapies. And, so it really falls on us to identify these patients. If you have access to these studies, and the SWOG study would be a great example, cooperative group, but getting them on studies. But as you also mentioned, access to these therapies is available through guideline sort of approval. And so I think don't feel helpless to not measure in the first place because there's nothing you can do, because there is something you can do in these patients.
Well, thank you very much, John. It's been a pleasure speaking to you. I think that we've outlined, an important aspect of treating GI cancer by testing for targetable pathways and especially HER2 overexpression. There are a number of different approaches to this now, and the exciting new data with antibody-drug conjugates. So we look forward to how we can combine these things in the right sequence and with immunotherapy to get the best outcome for our patients. And the best way that we're going to get the right data is through clinical trials. So as always, I like to encourage people to contribute to that and enroll your patients.
One more additional comment is it's been difficult to enroll the clinical trials because of the pandemic and staffing issues for all of us. So keep the faith, keep pushing. We will restore staffing to a level where we can pick back up clinical research around the country. So fingers crossed that we'll have that brighter day soon.
Thank you for joining us today. Remember, to receive your credit and evaluate this program, please visit morningcommutepodcast.com/solid tumors 6. Look for all of our podcasts on your favorite podcast streaming services.